I'm just going to make one announcement about the mingling session for today before we start the session. So the mingling today is actually going to be one session. It's going to be the same Zoom link, so where you got into the main hall, that's exactly where you'll be. The rest of you can all stay after the dancing, can come back in here, because the idea is what we want to do is have a discussion between the Israelis who are sitting here or on Zoom and our participants, our audience, that's sitting in the UK. So that's what the mingling session will be. And Professor Ilana Gozes, who just walked in, who you know, is going to be moderating that with some very significant questions. And we very much hope that many of you will participate with answers. Before that, though, and this is going to be at 14:20 Israel time, 12:20 UK time, a little disorienting, we're going to have the dancing, Dance the Moment, an interactive workshop. Those of you who are on Zoom will be able to actually see that. You can dance on your own at home or at work. And those who are present, please join us, and we're all going to dance. So I'm going to introduce Mellissa Barber, who's a graduate student in the UK, and she'll be chairing the session. Thank you, Mellissa.
Hi, it's my pleasure to introduce the next session on models of aging. And our first speaker is Professor Karen Steele, who is interested in genetics of deafness and pathways associated with age-related hearing loss. And she won the Brain Prize for her research in 2012. And she's also an elected member of the Royal Society. So today she's going to speak to us about deafness in humans and mice. Thank you.
Okay, thank you for asking me to talk today. I'll just find my talk and share the screen. Good. So I'd like to talk to you today about age-related hearing loss and the work that we've been doing towards understanding that over the past few years. First thing to say is that age-related hearing loss is really common in the population, and so it can start at any age and it affects many people. If you look at this figure on the top right, you can see that right on the top right, you can see this is the prevalence in the population of different levels of hearing impairment plotted against the age in years. And you can see that this blue bar represents 25 dB hearing loss, which is the level.
Sorry, Karen, if you can close the bottom of the slides, you have the next slides or something with the viewer, so we can see actually the full size of the frame. Yeah, thank you very much.
Sorry, is that better?
Yeah, it's okay. Thank you.
Sorry, I didn't do anything. I think you must have done something to sort it out at your end. Okay, thank you. Yeah, so 25 dBs hearing impairment is the level at which you benefit from wearing a hearing aid. And you can see that the numbers of people affected by these levels of hearing impairment increases until people in their 70s, 60% of people in their 70s have a hearing impairment, significant hearing impairment. So there's a lot of hearing impairment in the human population, and it's likely to be due to multiple causes. So it can be a mutation in an essential gene or exposure to noise, drugs, and infections. And I think there's probably going to be an interaction, well, we know in many cases there's going to be an interaction as illustrated in this bottom diagram here. So we can have environmental exposures that can contribute to a person's level of hearing impairment, and genetic susceptibility that can contribute to people's level of hearing impairment. And each individual person can be somewhere along this spectrum, along the bottom here. So they can have a mutation that causes a major contribution to their hearing loss, or they can have a very significant hearing loss due to a very loud noise exposure. But probably the genome is always involved in interacting with environmental damage, and also environmental exposures can affect people that have a genetic susceptibility as well. So people are somewhere along this spectrum. It's very difficult to unravel the causes of hearing loss directly in humans because we just can't get into the inner ear, and because of the numbers of people affected and the profound effects that it has on people, it's a significant risk factor for dementia, for depression, for social withdrawal. So it's a very high unmet medical need. There are no medical treatments for hearing impairment at all. So the mammalian ear, just a couple of slides to remind you of the structure of the ear. So we have the external ear canal, sound travels through the middle ear into the inner ear. This is the cochlea, the coiled part of the inner ear here. If you look at just one coil of this cochlear duct, this is the sort of thing that we see. The organ of Corti sits here on a flexible membrane that vibrates when sound is introduced into the ear. And there's this very specialized structure I'd like you to notice, which is called the Stereovascularism in purple here, which generates a very high resting potential and high potassium level within this fluid that bathes the top of the sensory hair cells, which are in red here. If we look down on top of this organ of Corti by scanning electron microscopy, we see this bottom right image. These are 3 rows of outer hair cells and 1 row of inner hair cells, and each individual hair cell has an array of modified microvilli called stereocilia on the top of them. The outer hair cells act as amplifiers of the sound coming into the ear, and the inner hair cells are connected to most of the efferent nerve endings, and so they are the primary receptors of the sound. And then if we look at this cartoon of the organ of Corti, there's an illustration of a number of different specialized supporting cells. So there's a lot of specialized structures within this organ of Corti to help keep the whole thing very rigid and able to detect sounds which are almost at the level of Brownian motion at the threshold of hearing. And one thing to note is that the sensory hair cells never regenerate during life. So once they're damaged and lost, they're gone forever. So how is sound detected by the organ of Corti? This flexible membrane here is deflected by sound waves coming into the cochlea. And when that happens, these inner hair cells and outer hair cells and their stereocilia at the top are deflected because they're held in place by this membrane, extracellular membrane here, and that helps to deflect these stereocilia bundles. What then happens is, this is a magnification of a stereocilia bundle here, is that deflection pulls on these tip links that join adjacent stereocilia, and they mechanically open or gate transduction channels in yellow. And when that happens, the potassium ions, which are very prevalent in this endolymph, which bathes the top of the hair cells, the potassium floods into the hair cell and depolarizes the hair cell, triggering synaptic activity in the neurons, which are in green on the left here. And the Stereovascularis generates this endocochlear potential, or EP, and it's required for normal sensory hair cell function because it provides a large driving force. When those transduction channels are open, there's a range from plus 100 millivolts to minus 70 millivolts, which means that the potassium ions enter through the transduction channels much more rapidly than they otherwise would, and that provides some amplification. So that endocochlear potential is essential for normal hearing. So a number of studies some time ago suggested that these are epidemiology studies and histopathology studies, suggested that there are 3 major sites affected in aging, and I think that more recent studies have supported that. So the Stereovascularis here is generally associated with aging and age-related hearing loss. And the sensory and neural defects, so sensory meaning the sensory hair cells, and also the innervation, the neurons innervating the inner hair cells, often are damaged by noise exposure, so they also contribute to age-related hearing loss. And it's because there are these 3 very different structures in the ear that can malfunction in some way, we really do need accurate diagnosis of the site of the lesion, and we're going to need to develop different treatments needed for different mechanisms of hearing loss. So in order to find out more about the molecular basis of these lesions, we turn to genetics. So I'll come on to that in a moment. But first of all, I'd just like to say we use the mouse extensively to understand the deafness, because we can look inside the inner ear at any stage, which is a great advantage in trying to find the early stages of the mechanisms of hearing loss, which is really important, which you just can't do in humans. So, so what have we learned from the mouse about cochlear pathology? And I've got just a few pictures just to illustrate the range of different pathologies that we can see. There's a very wide range of cochlear pathologies. So this is a picture, a high magnification scanning electron micrograph, a normal hair bundle or a stereocilia bundle on top of one hair cell. You can see the very neatly arranged V-shaped arrangement and 3 rows of tiered heights of stereocilia. And this is what you see in noise damaged stereocilia. Some of those taller ones have fallen off and disappeared, and others are bent. And we can have all sorts of different defects in different mouse mutants. So we can have long stereocilia here. We can have disorganized hair bundles here. We can have very thin stereocilia, and we can have very short stereocilia. So all of these features can occur in humans as well. And then to the neural defects, we can, this is just one example to illustrate, we can have swollen nerve endings. So these are a row of inner hair cells here, and these are, in green, are the swollen nerve endings underneath those inner hair cells. And that also is associated with the loss of the synapses. So that's one feature. And then we can have defects in the composition of the endolymph, or the voltage, the endocochlear potential. So either you can have a collapsed endolymphatic compartment here, so there's hardly any endolymph and it's not connected to the hair cells at all, or we can have a reduced endocochlear potential, as plotted here in these homozygous mutants, where the endocochlear potential should be in normal levels up here. And this is illustrates a normal Stereovascularis that is dissected out from the ear, labeled and laid flat on the microscope side. That's what it should look like, and this is what it looks like in some mutants that have reduced endocochlear potentials. So there's a variety, a very wide variety of different cochlear pathologies that can underlie hearing impairment. So the question we want to ask is also, is there a window of opportunity for intervention? And the reason we need to ask that question is that all of these different sorts of pathology that we showed you on the previous slide, they all end up with degenerated hair cells. That's a secondary defect, because all of these pathologies result in hearing impairment before the hair cell degeneration, but you end up with degeneration of hair cells. So there's only a couple of hair cells left here, and most of them have completely degenerated in this scanning electron micrograph. So this is a very common feature. So if we're going to intervene, we need to find that critical period in order to intervene. So just to summarize what I've said so far, there's a very wide range of cochlear pathologies that we see in different mouse mutants that are almost certainly going to be represented in the human population, if only we could look inside the cochlea at early ages. There are 3 key sites in age-related hearing loss. There's the stereovascularis, with the lateral wall of the cochlear duct can malfunction. The sensory hair cells themselves can be abnormal, or degenerate eventually, or there can be problems with the synapses of the neurons innervating the hair cells. Almost all of these pathologies lead to hair cell degeneration eventually as a secondary defect. And we need to ask, what is the window of opportunity to intervene in these situations? And diagnosis of the site of the lesion will be critical in order to stratify clinical trials to develop these treatments, and also eventually, when we have those treatments available, to guide the choice of treatment in an individual person coming into the clinic. So if I can move on now, in the second part of my talk, to talk about the causes of hearing loss. So we turn to genetics to try to understand the molecular basis of hearing loss. And so far, there is extreme genetic heterogeneity. We maintain, or Morag Lewis in my group maintains a manually created list of Deafness genes that are known either in the mouse or in humans. And we have 832 on that list so far. So a lot of them have only been described so far in the mouse as causing deafness in the mouse. Some have only been described in humans, and there's a growing overlap where those same genes are involved in human and mouse deafness. Now, almost all of these were found through rare human mutations by positional cloning in families, or by mouse mutants with developmental defects causing deafness. But these same genes are also good candidates for age-related hearing loss because we know they're involved in normal hearing processes. And each gene has a different role in hearing, so you can imagine over 800 different ways that we can develop hearing loss. It's very heterogeneous indeed. But the reason it's important is because genes give us clues to the underlying molecular pathways, and that helps us to develop treatments, whatever the trigger is, whether it's a noise damage or a primary genetic cause. The same pathways are going to be involved in that pathological process. And many of these pathways have existing drugs already, so we really just need to find out more about those molecular pathways involved. We also know that there are going to be further genes that we haven't yet discovered, and this is especially true in age-related hearing loss. So can genetics give us clues to the genetic landscape of age-related hearing loss? Because often there's a common hypothesis where there is a common disease is due to common variants in the population that all add up to contribute towards the pathology. We really just don't know if that's true in hearing impairment yet. So how can we detect these genes and pathways involved in age-related hearing loss in order to develop treatments? So an understanding of the genetic landscape will help a lot. So just want to take one example from published literature already. If we look at those genes that are already known to be involved in human dominantly inherited deafness, these are called the Dfna genes. There are 44 genes that have been identified in humans so far. Of those 44, 39 of them lead to progressive hearing loss after normal early development. So the person develops normal hearing and often normal speech, and then at some point later in their life they develop the onset of hearing loss. And the hearing loss in these families can show a wide range of ages of onset, from early childhood to as late as 60. So this suggests a priori that rare single gene mutations can also contribute to age-related hearing loss in the wider population. These are just a few that we've identified so far. So what we've done here is plotted just a handful, 5 or 6 different well-known deafness genes to illustrate the fact that these genes are already known to be involved in both common hearing loss in the population, as well as rare cases of severe hearing loss. And what we've plotted is the effect size, so from an association in the population through mild up to profound hearing loss, against the minor allele frequency, so how common these variants are in the population, how common these mutations are in the population. And every gene that has been that's marked on here has been shown to be involved in common variants in the population, but also that same gene can be involved in profound hearing loss or severe hearing loss in individuals in the population. And recently published genome-wide association studies, which I've indicated down here, of age-related hearing loss, have reported the association of several known deafness genes with hearing loss in the general population. And here's some of those genes that have been reported in those papers. So it looks as if there's going to be an overlap in the genes involved in hearing loss in the population, age-related hearing loss in the population, and also childhood deafness, early childhood deafness. So we've been doing some exome analysis of well-characterized cohorts, a Charleston cohort, which are adults over 55 years old. We have 450 people with hearing loss and also a group of 78 people with normal hearing. And we've looked, this collaboration with Judy Debner and Brad Short in Charleston, we've looked at both common and rare variants in these exomes. And I've just got 2 data slides to show you to finish up my talk. First of all, looking at common variants that presumably have a small effect size on the protein. We ended up with 450,000 variants in this population to analyze, and we've looked at an outlier analysis and a regression analysis, and that's what we've plotted here. So this is a sort of burden test, a variety of burden test, where we're plotting for each point, we've plotted the number of variants in that gene in the group with hearing loss against the number of variants in that gene in people with normal hearing. And all of these are circled in orange as significant outliers, suggesting that there's a higher burden of mutations in the general population in those genes. And there's 167 outlier genes in people with hearing loss. But surprisingly, we also found a number of outlier genes in blue in people with normal hearing, which is not what we expected. There's a few genes known to be involved in deafness, but there's only 6, so there's not very many. And that suggests there's going to be a number of quite a lot of new genes that we need to follow up to establish if they do really underlie age-related hearing loss. So that's a burden test in the whole population. And the last data slide is looking at rare variants with large effect size. So in this case, we included variants predicted to definitely affect protein function because they have a high score in the variant pathogenicity predictors. These are all rare in the population. We had 26,000 variants to analyze, and we've plotted the audiograms. So this is a sort of a variety of Mendelian randomization where we had 5 or more people with exactly the same variant, so not a gene, but a specific variant. We plotted the audiograms for those people compared to the rest of the group. And 2 examples, CAPN9 here, this rare variant led to worse hearing. So this is a standard audiogram for humans. The green represents the normal group, the rest of the group, and the blue represents people carrying that particular variant. And they have significantly worse hearing. This is the left ear, this is the right ear. So that variant does appear to be involved in hearing impairment. But this variant here in TCENC2 seems to give better hearing, which again is a surprise. So better hearing compared to the rest of the group. Okay, so we do think that this suggests that rare variants can contribute to hearing impairment. So just to summarize, hearing loss can be caused by a single mutation in one of over 800 known deafness genes, and that number is increasing all the time. The mutations in genes known to underlie childhood deafness can also be involved in age-related hearing loss. Some variants are associated with better hearing, which is not what we expected. Hearing loss, this is the question, is hearing loss also due to combined effects of genetic variants in a number of different genes? This really needs further study. We're looking into that. And we also know that the environment interacts with the genome. The message I want you to go away with is that there's a very high level of heterogeneity in both the genetics and the cochlear pathology that we see in hearing loss. And that suggests that we will, in developing treatments, we're going to need to focus on key molecular pathways or types of cochlear pathology or sites of lesion in order to develop cost-effective treatments, because there are just so many different genes involved. And I'd like to just finish by thanking the people who've contributed to the data I've described today, mostly Morag Lewis, New Lingham, Jung Chen, people who were members of my group contributed to the phenotyping, and also to our collaborators, Judy Dfna and Brad Short. And thank you to all the funders of this work over many years. So thank you very much.